Hello everyone, my name is Vaidi Kepalla, and today I want to explain one of the recent imaging techniques which is called magnetic particle imaging, and I will explain about the different application of this method. And this is outline. At first I explain about the introduction of magnetic particle imaging, and then I demonstrate how this method works, and I also compare this method with current methods in imaging. And at the end, I mentioned the application of MPI methods such as cell tracking, tumor imaging, and magnetic hyperthermia. So this method actually was invented in 2005 in Germany by Gleich and Weisenegger. The principle of this method is locate and quantify the tracers in specific areas. So MPI could scan the electronic movements of superparamagnetic iron oxide particles, which is 22 million times more intense than MRI. The MPI characteristics is such as linear, quantitatively high sensitivity, and high spatial resolution, which is approximately 1 millimeter, and it has a potential to reach more than 300 micrometer. And there is also no background signal from biological tissue. So actually, the MPI method have two main parameters. One of them is superparamagnetic iron oxide nanoparticles, and the second one is the MPI device. Now here I explain about the superparamagnetic iron oxide nanoparticles. They are FDA approved and they don't have any radiation. And also these nanoparticles are designed to clear through the reticuloendothelial system. So it shows we don't need to worry about that, and also it's very have a good features for patients have a kidney disease. And this nanoparticle have two important for optimization. One of them is the core of the superparamagnetic, which controls the MPI properties, and the second one is the coating, with which material cover these nanoparticles, which affects the behavior of particles and in vivo and interact biologically. And this surface coating can be enhance cellular uptake or to increase the blood pool half-life. And also the best single core of these tracers of these nanoparticles are about 20 to 25 nanometer. And in this video, you can see when we apply the magnet in the liquid which has nanoparticles, then these nanoparticles are appeared and we receive a signal from them. Now I explained the theory of the MPI method. Here there are lots of magnetic needles, and when we apply the magnet on them, they are interacting and they are spinning. So it shows they are not saturated here. But we use a trick. We put outside in a magnetic field, which the n polar is according to the right figure is toward to each other, and this feature, this act, it's resulting all the needles are saturated except the middle one. So it means when we apply the magnet from the outside, except the middle one, all of them are saturated, and we just receive a signal from the middle magnetic needles. And now we demonstrate how the signal are produced when the field free point and the nanoparticles are interacting with each other. At first the nanoparticles are desaturated, and then by the crossing of the field free point on them, they are flipping and produce a signal, and then they are resaturated, something like this. And you can see when the field free point cross the nanoparticle, they are flipping and produce a signal. Okay, here we could see the line of the nanoparticles, and when they cross the field free point, they flipping, and by flipping of them, we could see the signal produced, and then they are resaturated. And in another video, you can see the nanoparticles in a letter C, and when they cross from the field free point, they produce a signal. And the signal is converted to an image, and you can see the letter C, the image of letter C here is appeared. Here's the comparison between the MPI method and other modalities. In fact, MPI image is straightforward, and MPI has a positive contrast. Unlike PET and SPECT, it is a tracer modality. On the other hand, we have an optical method, which is regularly used in research lab, but the problem is it's not quantitative. And the SPECT image is also clear, but the problem is the tracer only lasts for 2 days, and then the tracer decay, and we cannot track the cells. And also, but in MPI tracers, which reveals the pathophysiology with zero radiation, and also they have a 3 months persistence. So the first MPI application in preclinical research is mentioned to a cell tracking, like stem cells, cancer, and immune cells. Now the question arises, why we need to track the cells? And in this case, why we need to track the stem cells? One of the important stem cells is mesenchymal stem cells, which is used in tissue engineering. You suppose a person have a pain in the knee because of the articular cartilage damage. And when we inject the MSC cells to the knee, it's resulting to improvement in the articular cartilage damage. But unfortunately, sometimes these MSC cells undergo apoptosis, maybe because of lack of nutrients. So this apoptosis could attract the immune cells like macrophages to the implant site, and this resulting stem cell rejection. Therefore, we need a technique to quantitate the MSC engraftment over time. And if we find the answer, it will be so precious for cellular therapy. Another reason is, according to some papers, to some histological reports, the researchers find out the MSC cells could trap in the lungs, so it has some side effects for the body. And another reason is tumorigenesis of MSC cells. Actually, my research direction is about the cancer immunotherapy. So it's really important for us to know, after we inject the MSC cells to the body, what's the fate of the MSC cells? Do they degrade it in the body, or maybe they transfer to the tumor or metastasis? And here Bujeng designed a project, and in this project he cultured about 5 million MSC cells, and then they add the Rezovis is a type of magnetic nanoparticle to the cell culture. Then these stem cells uptake this nanoparticle. Then they inject this labeled MSC cells to the various mice. At first, when they inject these MSC cells to the mice, immediately they stuck in the mice, but they do not permanently stuck in there. After that, they went to the liver, and in that organ, they biodegraded. And this is the picture. It shows when they inject only the magnetic nanoparticle to mice, and they do not stuck in the lung. They directly went to the liver, and they biodegraded in that place. So this shows MPI has a unique advantages in cell tracking because it has a high sensitivity and high resolution. And secondly, the these superparamagnetic are relatively small and can be easily uptaken by the stem cells. And this is a video. This is actually the three-dimensional image of the MSC cells which stock in the lung of the mice. Another research which is done by SEAL in Western universities related to MSC tracking, but in this study they used three different methods, three different modalities, such as ion-based MRI, fluorine-19 MRI, and MPI cellular imaging to monitor and quantify the persistence of transplanted MSC and after that the infiltration of macrophages in vivo. Actually, these three modalities are complementary and provide further information such as specificity, sensitivity, and quantification of cell number. Moreover, in this study, they used ferumoxytol for labeling and detection of the MSCs. Actually, in this study, the MPI signal have a linear relationship with iron oxide tracers, and this provide the opportunity to quantify accurately the superparamagnetic iron oxide labeled cells. And here are some histological reports from the MSCs which labeled with ferumoxytol. Actually, these histological reports are stained with Prussian blue, and in figure A, you can see the MSCs uptaken the ferumoxytol, but in figure D, these are the unlabeled MSCs. In figure B, you can see the ferumoxytol labeled MSCs, and when the number of MSCs is increasing, actually the iron content and the signal we're receiving from the MPI is also increased. And the figure E is actually our control and shows when the MSCs are unlabeled, we didn't receive any signal from them. And this figure demonstrates the three different modalities. The figure A related to proton MRI, which shows the which shows the MSCs in the place of administration. And the second one is fluorine-19 MRI. Actually, the PFC positive immune cells are detected in the bone marrow, lymphoid nodes, and in the muscles surrounding the MSC implant. The figure C shows the MPI method. You can see two bright spikes, the smaller one related to ferumoxytol positive MSCs, and the big spots related to iron's food in the gut. So actually, you can see in the day 12, these MSCs are disappeared because they are degraded in the liver. But another spot is exist because it's related to iron food. So this is one of the potential limitation of the MPI method because they need to solve this problem to prohibit from the intervention of these spots. And this slide related to histological validation. The figure A and B are hematoxylin and eosin staining. A is 10 times magnification and B is 40 times. And the picture C, the black arrows refers to the mesenchymal stem cells. And finally, the picture D, the black arrows related to infiltration of macrophages in the place of MSC cells. So in my opinion, in tissue engineering, these macrophages may be result in stem cell rejection. And in terms of using of MSC cells for the purpose of cancer immunotherapy, maybe this macrophages, for example, the tumor-associated macrophages result in tumorigenesis or metastasis in cancer. And here is a brief comparison between different methods in tracking of cells. The first one is histology. Actually, the histology provided detailed molecular and morphological information, but they are just limited to a single time point and portion of tissue. In fact, we cannot survey the cells during the time and in vivo. The second one is MRI. MRI also is a non-invasive for tracking the cells, but the problem is you can see in the figure C, the distinguishing between the superparamagnetic and the air is really difficult. And another one is optical or bioluminescence. The bioluminescence is also have a good sensitivity and it has a wide range of proofs, but the cells which are in a deeper tissues, it's really hard to track them. And but in MPI method, it has a positive contrast, and we can receive the signals even in deeper tissues. And the tracer also doesn't have any radioactivity in comparison with the PET method. And slides shows the examination of depth penetration between the MPI and bioluminescence method. In figure A, you can see when we go into a deeper tissues, it doesn't affect the MPI signals, but in figure B, in fluorescent imaging, when we go into a deeper tissues, actually the signals are diminishing. And in figure C also shows a graph which shows exponentially decreasing of signals from the optical method when we go into deeper tissues. And it is believed more than 90% of cancers are curable if we diagnose them in early stages. So it shows the importance of the imaging method, and if we could image them in early stages, with a quantitative method, the physicians could cure the patients immediately. But how we can connect the MPI method with the cancer? Actually, at first I want to explain a phenomena. Its name is enhanced permeability retention. It mentioned the tumors actually have many vessels, many leaky vessels, and these leaky vessels provide the opportunity for the nanoparticles could release within the vessels to the tumors. So the Connolly at the University of California used the magnetic nanoparticle as imaging tracers, so because these particles accumulated in the tumor by the EPR effect. So when the nanoparticle went to these tumors, they could, by using of MPI method, they could diagnose the exact place of the tumors. Of the Connolly experiment about the tumor imaging. At first, they implanted the tumor to a nude rat, and then they injected tracer about 3 milligrams through the tail vein, and then they imaged the rat about 4 days. So here you can see after the injection, the tracers are accumulating in the tumor sites after the 6 hours, and then they degraded from the body. And here's the three-dimensional image from the mice in the time of 6 hours after injection. Actually, this method also could help us to determine the stage of tumor. The third application of the MPI method is when they combine with magnetic hyperthermia therapy. In this video, I explain the concept of the magnetic hyperthermia. You can see here the nanoparticles are lined up with magnet, and when we apply the alternating magnetic field, they are moving very fast. And if we increase this the moving of the magnet, the nanoparticles are going to be more hotter. And so this gonna be helpful for killing of the tumor. Here the doctor inject the tracers, inject the magnets to the body of the patients with the cancer, and then according to the EPR effect, these nanoparticles going to within the tumor size, and then the physicians apply the alternating magnetic field on the patients. So these magnets you can see are moving fast and going to be hotter and hotter, and resulting the shrinkage of the tumor cells. Here's the thing: how we could target the tumor precisely in purpose of treatment and in the same time image it. The answer is the combination method with MPI and magnetic hyperthermia therapy. So in more details you can see, according to the paper which is published in 2018, at first they injected tracers to a mice which has a tumor, and then you can see the traces going to the tumor and the liver, and then the operator select the target, and then with the MPI method, which has a field free point, select the target, and increasing the temperature by applying the alternating magnetic field, the tumor will be shrinkage. So this brings a novel characteristics for this combination method. It shows not only we could diagnose the tumor with this method, but also we could treat it. So here's there are more information about this combination therapy. Actually, here is a normal situation after the injection of the tracers to the mice. And in second image, you can see the AMF, alternating magnetic field, is increasing, and the temperature of the all organs such as tumor, liver, and rectum is also increasing. In the third picture, you can see by the fifth free point, they target the tumor and see just the tumor temperature, the red one, is increasing. The other organs, because the magnets on this organ are saturated, the temperature is not going to be higher. And in the last picture, you can see when they target the bottom tumor, only the temperature of bottom tumor increasing, and when they target the top tumor, only the temperature of top tumor is increasing. And in conclusion, magnetic particle imaging by using of field-free region could locate and quantify superparamagnetic iron oxide nanoparticles, and also give us an insight to non-invasively monitor the fate of cell population in vivo. And this could enhance therapeutic monitoring to confirm appropriate MSC delivery, measure the number of MSCs present over time, and quantify immune infiltration to identify MSC rejection or tumorigenesis. And combination of MPI with magnetic hyperthermia not only result in diagnose the cancer, but also we could treat them. And the limitation of this method is loss of signal in MPI images because of MSC apoptosis or clearance or during the proliferation of them. And also this application is not applied yet on human model. And these are the references, and thank you for your listening.